As we saw in the replication, DNA polymerases has a problem. They all can do only 5' to 3' direction synthesis and not in the reverse. And also they have another problem. They cannot start de novo synthesis. And the de novo synthesis was that DNA polymerase requires a primer to start the replication. This creates another problem, the end replication problem for the DNA. Now we have the linear chromosomes. In the circular chromosomes, like in bacteria, this problem do not exist. This is only true for the linear chromosomes. Now I will write the directions. First I will go from inside. This is the primer. It goes like this, extends, and this is the primer. And it goes like this. Here, 5, here. There are multiple primers because there are multiple origins in the chromosomes. They're not this close in the chromosome, but still for the this is just for us to understand how the replication is done. So you have to extend this to a larger chromosome. And these are the RNA primers. What happens is the polymerases then come and erase these RNA structures and replace them by DNA structures. This is what happens normally in the replication. But there is another problem here. The problem is this is the 5' end. This is the 5' end. But in here, we will have a primer here, and we will have a primer here. Now it extends this one. It extends this one. This one is going this direction. This one is going this direction. Now, if it erases again the primers, it can refill this one, refill this one, but because we don't have a primer in here now, it cannot start a synthesis from here. And it cannot start a synthesis from here because there is no primer. And it cannot extend from here to here because it will be 3' to 5', which is the opposite direction of how the DNA polymerases work. So, in each cell division, the telomeres are shortened by 20 base pairs. 20 base pairs is the average length of a RNA primer. If there were critical genes at the ends of the chromosomes, for example, these are the chromosomes and these are the ends. Here we have A gene, here we have B gene. So after a couple of rounds of cell division, we would have lost the genes A and B. To prevent this, the cell has structures called telomeres. And the telomeres are present at both ends of the chromosomes. This is telomere. It is usually wrapped up into heterochromatin. And in order to prevent the spread of the heterochromatin, there is a barrier in here. This is the barrier structure. So the heterochromatin do not go in here. So there are genes at this point, and the genes at this point are protected by the chromosomes. So each division, the telomere gets shortened, and the telomeres do not code for any proteins. They are just repeat sequences. They shorten each time, and whenever they are shortened at a certain point, then cell cannot divide further. This is called the Hayflick limit. Hayflick is the name of a scientist. This is called the Hayflick limit, and each cell in our body, somatic cells, have a lifespan. Like, for example, a cell can divide 60 times and then should stop dividing. What happens when a cell stops dividing is that it doesn't die, but it gets into the G0 state, which is a resting state. And G0 state, actually neurons are always in the G0 state. But for example, your epidermal cells divide much more than that, because you lose all of your skin in every other couple of months, for example. They have a higher limit, they have a higher number of cell divisions, for example, but eventually they also stop dividing. That's when the wrinkles or creases appear in our skins. So it is called, as you might expect, this is called the aging. The Hayflick limit is directly related to aging. For an organism to age, its cells have to age, so this is called the cellular aging, or cellular aging is called senescence. Senescence is the cellular aging, so it depends on the telomere, but it has other versions, so it is usually named like telomere dependent senescence type of. Why the cell stops dividing is that when the telomeres reach a very short length, then the DNA repair system sees this as a broken DNA, and it alarms the cell to go into DNA repair. And because the DNA repair system recognizes this as a chromosome abnormality, then it forces the cell to switch into the G0 state. There is also the telomere has a special shape, and it contributes how telomere has a special shape, and it contributes how the DNA repair system recognizes it. Usually, if this is the chromosome, telomeres are found like this. This is called telomere. These are the telomeres, but what do you call them? T-loop structure. Telomere is found in a T-loop structure, so that the cell or DNA repair system will not find this as a broken end or something, and because it is getting shortened each time. For example, this part is missing, but the DNA repair system will not see this as a break. But when it's in a short, when it is shortened, then it cannot form the T-loop structure, so that the DNA repair system comes here and sees this as a DNA break and directs the cell into the G0, because DNA repair systems will repair like missing bases or substitutions or timing structures, but a DNA repair system do not repair a broken chromosome. Chromosome instability or chromosome aberrations are not to be repaired. It is much more easier to let the cell go and produce a new cell. So, but in cancer cells, for example, because DNA repair systems are not working, there is a lot of chromosomal instability or chromosome breaks, for example. So, this is held into T-loop structure, and also in the T-loop there are lots of proteins bound to the telomeres, and this is called the shelterin complex. Shelterin complex attaches to this T-loop structure and holds this in its shape and hold it stable. Also, the telomere is usually out of the cell cycle, is wrapped into the heterochromatin. Now, why does it now the telomeres get shortened in every cell division, in every somatic cell of our body. But this is not true for our gametes, like sperm and egg cells, and also it is not true for the stem cells, for most of the stem cells. Because there is an enzyme which maintains the telomeres, and it is called telomerase. Telomerase is active in the embryonic stage of humans, and then in the first month, maybe after birth, the telomerase gene expression is shut down. So we have to continue our life with the telomeres that are long enough for us in the beginning of our lives. And the telomerase cannot become active in our somatic cells. If it becomes active, it means it's a cancer cell. Cancer cells, most of the time, reactivates the telomerase so that the cell do not go into the G0 phase, and so that it can continue adding nucleotides to the telomeres. Before we are born, in the gestation period, the telomerase continues synthesizing the telomeres so that the organism borns with longer telomeres. There is an average telomere length for humans, for example. But there is a disease, this keratinosis congenita. These children are born with shorter telomeres because during the gestational period, because of a mutation and TERC gene, I will explain in a minute. Because of a mutation in the TERC gene, these children are born with shorter telomeres, so their telomeres get shortened at the age of 10, like as it would be at the age of 90. So at the age of 10, they look like 80-year-old person. It's called accelerated aging. And so the telomere length the organism starts its life is critical. And this is also proven with a very recent study, which I will mention in a minute. The telomerase is encoded by TERT gene, and there is a telomerase RNA component, and it is called TERC gene. The telomerase is an RNA polymerase, RNA dependent DNA polymerase, as it is in the reverse transcriptases, and it uses an RNA template to synthesize the DNAs. It serves as a primase in its own, but it comes like a huge structure. It comes as a huge structure. Suppose that this is the telomeres, and the cell maintains its telomeres. During the gestational period, the cell divides a lot, so its telomeres should be at the maximum level before birth. So telomerase has to replace the missing parts of the telomere. And in here, this is the RNA part, protein part. It comes here and synthesizes a DNA here. So that by doing so, it extends the telomeres. By doing so, it provides a primer for the DNA so that it can start the replication from there. And so the RNA serves as a template. The structure in the telomeres is not random sequences, but it doesn't code for anything. The human, it is GGG, TTA. It's 6 nucleotides. This is repeated after other GGG, TTA, and it is always repeated. So the RNA primer here is the complementary of this, so that not complementary. The RNA component has another structure. It comes and serves as a primer so that DNA can be synthesized, but then the part which is synthesized from the RNA will be degraded, for example. And in the gaps, again DNA polymerase synthesizes the gaps as usual as in the other parts of the chromosome. So in each cell division, again there will be some loss of chromosome, but this time it will be lost from the new sequence that the telomerase is added. So if the cell divides 100 times, the telomere length will be constant in that cell. The polymerase is the book says, works like this. These are the DNA and RNA structure, for example. The polymerase holds it like a hand, and in the palm, they say, in the palm, it is the catalytic region which does the synthesis, but in the fingers and the thumb is required to hold the DNA together. Like if you remember, DNA polymerase was a hollow enzyme complex, but the telomerase is always in a compact structure, so it works like this. I said the chromosomes get shortened 20 base pairs, but it's like 100 to 200 base pairs in each cell division actually. So this is how a cell the cell gets shortened each time it divides. Telomere length gets shortened when an organism ages, and there are no exceptions to this rule, and nobody is exempt from the cellular aging. Only cancer cells do not age, and they are immortal, and they have telomerase activation in their cells. There is a direct correlation between age and the telomere length, but there is a graphic will tell a lot about how it is correlated. Now this is done by a company, QIAGEN. So this is here telomere length, and here there are individuals at different ages. Zero age means cord blood, and then it goes up to 100 age. They even included people at age 100, and they recruited like 2,500 people in this study, so it's a huge study. And the telomere length shown like this, so it declines linearly with the age, and it is highest at the age 0. But the actual data is like this. This is a trend line, if you know what I mean. Trend line is the optimum line that connects the dots. For example, if there's a dot here and then there's a dot here, the trend line will go here. So the graphic was something like this. And you have to imagine this for the 2500 people. I think this should be enough. So it declines with age, but still it has variability among individuals. What it means is that, for example, this person is 20 years old and this person is 80 years old, and this person has longer telomeres than this one. Why is it so? It has to do with the length of telomeres that you start your life, and it varies between people. So some people age earlier, for example, and this person, for example, has a very high telomere length, but not as high as this one, the 0. So there are not very extreme cases, like at the age of 10, for example, if you see a telomere length like this, it means that this is not normal. This is actually dyskeratosis congenita syndrome, because they have started their life with very short telomeres. Or if you have a 90-year-old with telomeres like this, it might mean that you get the sample from the cancer cells, for example. So there are mathematical data in here. The R square for this graph is 0.4642. You would want this to be closer to 1. The ideal one is 1. Ideal situation is 1. But 0.4, it means that there is high variability between individuals. Usually this number is not good for a publication, for example, because they will say that your data is like everywhere. But the author says, although the R square is like this, p-value is 0.00001, so it is highly significant. It is because this data is made up of 2500 people more than. If you did this like from 10 people, individuals, then this won't be significant and this will look not good and it won't be accepted. But this represents the public, for example. So this, I like this graph a lot. In the previous graph, we saw that the telomere lengths are variable, but you can't like say this. You can't say like this person has longer telomeres than this person, so this will live longer. You cannot say this. And there was a huge debate about why telomere length is not correlated with the lifespan, which I mean, mouse has very long telomeres, but it lives only 3 years, and human has relatively shorter telomeres and it has a 70-year lifespan. So the dilemma is solved by Blasco et al. This is a 2018, I think, study. They found that what determines species lifespan is not the telomere length at the start of the life, but how fast the telomeres get shortened. So we used to think that because the mouse has longer telomeres and human has shorter telomeres, so the maybe mouse does not have a telomere Hayflick limit, for example, and it after 3 years it dies, but it still has lots of longer telomeres. This study says it's not like so. The rate of the telomere shortening determines the lifespan, means this. For example, humans telomeres get shortened 70 base pair in a year. like this is the average, because you have neurons not dividing, gut cells are dividing, so this is the average number. Elephant 110 base pairs, and when you come to mouse, its telomeres get shortened 7,000 base pairs per year. So this is a huge number. And then they did a graph of lifespan versus rate of telomere shortening, and they found a correlation. So human has the highest lifespan, but it has the shortest rate, and the mouse will be here, has the lowest lifespan, but it has the highest number of highest rate of telomere shortening. And they did this not only in 4 animals, in lots of animals they measured the telomere shortening rate, telomere lengths and their lifespans in mouse, goat, gull, reindeer, vulture, dolphin, flamingo, elephant, and another 7 or 8 animals are added. So this, I think, is a very important finding, and yeah, it explains the lifespan and the telomere length dilemma for us.